Oh, let's get rid of that little thing. There we go. So these excited electrons, just like you guys, so excited. From that photosystem 1, which is, remember, the second photosystem, taking that NADP plus, and adding that hydrogen, powering that to give that hydrogen to turn it into that energy-containing molecule. Both of these, remember, go to the Calvin cycle. Out there, and they're in the stroma already, as you can see, and then they head on over again, checking out all those oxygen. Oh, we breathe it. Hydrogen is getting pulled in, remember, inside the thylakoid space. And these hydrogens then moving out through ATP synthase, making that ATP. All right, so these are all, we're going to go get into a whole bunch of different molecules that are involved in the Calvin cycle. Probably the big ribulose biphosphate, RUBP, is kind of the main molecule that we see. Carbon dioxide, you know, this is our carbon fixation step, where we take the carbon from the air, we turn it into a chemical product, you know, in the form of sugar. Catalyzed by this enzyme Ribisco, which stands for ribulose biphosphate carboxylate, which is RUBP. Let me write it out. Ribulose biphosphate. Remember, I know I have some chemistry kids in here. Carboxylates. And this is just showing that at the end of it turns into the G3P molecule. We'll see that in a second. But here, that first step, we got CO2 entering this RUBP, creating glyceraldehyde phosphate, or glycerate 3 phosphate right here. And this first fixing that carbon from the air chemically into a molecule that can then move on through this this cyclical metabolic pathway that we have here. And here, that another reduction oxidation reaction. So we're reducing this G3P and we're making it triose, a TP, triose phosphate. So this is getting oxidized. And we can see, you know, this process is using this NADPH molecule, so it's getting oxidized because it's losing a hydrogen there. So if this is getting oxidized, you know, this other molecule is working on it's being reduced. Confusing. But here we have our triose phosphate, so we're about halfway through the cycle. We see that right here. And this is where we're about ready to make a little bit of sugar. So that's what happens here. Some of that TP is linked to form this glucose phosphate. And that then can go on to make starch, you know, gluing those or connecting those molecules. Remember, condensation reaction, there's water as H2O as a product. The way I think of, like, what is condensation? I think of condensation on your car window in the morning, because you've all seen that, where water has changed from a gas to a liquid on the windshield. So I think of condensation, I think of water, making water. That's what's happening here when you're building those 3 and 6 carbon molecules that are being produced by the Calvin cycle. So there's one more step. We have to add energy here to recreate this RUBP from our G3P. So G3P rearranged, phosphorylated, and that's coming from that ATP. Okay, phosphate is added. Regenerate that RUBP, and remember that G3P was removed from the stroma. We'll say shuttled out means removed from stroma. Where your body then takes it and builds whatever molecule it needs. The body is amazing. It can take, your cells can take all these different molecules and break them up and rearrange them. All right, so structure and function. We got double membrane. That's what something I mentioned earlier. Symbiosis, meaning it probably, well, it did exist on its own before, and chloroplasts it's not in this diagram, but they do have their own DNA and their own ribosomes. We don't think about that so much. We talk about it a little bit with mitochondria, having that basic machinery that all cells need, but so do chloroplasts, and they use them sparingly, but they are there, which indicates, again, it probably was an independent cell at some point in time. So electron transport chain, synthase for our photophosphorylation. I kind of fit it. These random, you'll notice they're kind of flat, and they don't have a large interior. They're kind of squished down. That's to give them a nice to increase the surface area to volume ratio. So man wants that surface area to volume ratio high. Most of these cellular machinery things in your cells in general, we need a high surface area to volume ratio. Okay, it's just surface area volume ratio, you want it high. And in this case, and just like in the intermembrane space in the mitochondria, when a similar thing was happening, when hydrogen was being pumped across the membrane, we want to increase that concentration quickly, so quickly accumulate ions. If you have a large interior surface, a large interior volume, that is, my apology, it's going to take a lot longer to build up that high concentration. Remember, just see how small that volume is? You don't have to shove too many hydrogens in there before you get a high concentration. And here, let's apply this. The lollipop apparatus. So cool. We'll see a picture of it in a second. Remember, it uses that radioactive isotope carbon-14, and that makes basically radioactive carbon dioxide, because that carbon is incorporated into the carbon dioxide, so it can be traced. This is all to make it traceable, because you can take an X-ray image of it and it'll show up to make traceable. And we'll see that in a second. So they supplied this CO2, this radioactive CO2, to this algae, and then took samples at short time intervals and killed them. They killed the algae, sampled them, and then they put them in hot methanol, and that fixed the molecules, and then they basically separated them using chromatography, autoradiography. But yeah, just looking at here, there's light, there's the lollipop, you see the algae growing in here, removing them, putting them to hot methanol, and then they end up putting them on paper. And you will do this shortly, some chromatography. It's pretty fun, I think. So yeah, actual picture. Look at this. It just looks beautiful, this green. Anyway, we see the algae killing them at those time intervals. I believe it was 5 seconds and 30 seconds. And we'll see in a second. Oh, you see this is the part where the radioactive carbon-14. This is all the algae. Nice. See how it almost looks like a thylakoid, right? Large surface area. The light can come through. It's like on both sides. It's like some kind of filter there. I'm not sure what was going on there, but they take that green algal that solution. They do a thing called spotting where they build up the chlorophyll, the metabolites, whatever they get coming out of the lollipop, and then they allow, usually it is some kind of solvent, like it's like almost like gasoline. Solvent can be water, acetone, alcohol is used, and then it's a type of hydrocarbon. It's like it's a little more refined than gasoline, so it's very flammable. We don't like to use it too much, but that stuff works the best. But we don't want to light on fire. Gasoline, that stuff is really flammable. Okay, so yes, 5 seconds was this first. They let it photosynthesize for 5 seconds. Boom, took algae out, killed it. And then it say, hey, what's here? And they saw that PGA, this glyceraldehyde, this G3P basically, was that first product in carbon fixation. And then they went another 30 seconds and got these other these molecules that we've mentioned. So all these, they show the intermediate and final products of the light-independent reactions. Yeah, so we will see our chromatography won't quite look like this. It'll look a little different. But that's one way you're basically separating, using the paper to separate the larger from the smaller molecules. So I need you to draw this. You'll see on the next slide. Draw and label a chloroplast similar to this. Make sure you have a granum, a stack of chloroplasts. You have the envelope, which is basically the membrane surrounding the chloroplast. You see this is the stroma, just the empty space here, and then very right here you can see the thylakoid membrane. So extra points, you color it really nice and label it up. Have fun with it. I give extra points for those of you that like to flex your artistic skills, okay, that you have honed over the years. So all right, have fun with the diagram.